We move now to the final concept in our unit looking at carbohydrates. This concept is studying a property of some carbohydrates known as their ability to reduce other species. Basically, if a carbohydrate sugar has this unique ability, namely to reduce another species, we call it a reducing sugar. If a carbohydrate does not have the ability to reduce another species, we say that it's a non-reducing sugar. Now that sounds complex to start with, but it is very straightforward, and it's related to the introductory work you did in Year 11 on redox reactions. Let's take a look. So thus far, sugars or carbohydrates have been introduced as ringed structures. In other words, their structural formula is best represented by a ring. Now this is only a half-truth. In solution, the ringed structure of many carbohydrates can open out into an open chain. If you'd like to think about it as a padlock, for example, is a closed ring when it's locked, but if you insert the key and open the ring has been broken and no longer is cyclical. That's true of carbohydrate sugars that are called reducing sugars. Now, this opening and closing of the chain in solution is in equilibrium. In other words, the sugar will continually close and open. Even more interestingly, the opening and closing of the chain can result in either an alpha or a beta form of a sugar regardless of its starting structure. This means that you may start with alpha glucose, it will open up into a chain in solution, and then reclose back into a cycle that may well form beta glucose. This is known as alpha and beta isomerism. Just pause for a moment and see if you can remember a definition for an isomer. Hopefully you remembered a definition along the lines of a molecule with an identical molecular formula to another molecule, but with a different structural formula. Now this constant changing in solution from alpha to beta forms is the driving force behind reducing sugars and is called alpha and beta isomerism. Let's take glucose as an example of an open chain form. Here's your ringed structure of glucose that you're used to identifying. Methanol group at the top of the ring, hydroxide group on the opposite side. Alpha is opposite, so we have alpha glucose. In solution, this ringed form of alpha glucose is in equilibrium with an open ringed form. Please note the hydroxide group has moved down lower, a double bond has formed to the oxygen, but the chain is split from the ringed oxygen. You now have an area of reactivity at the double bond. This slightly kinked form of the broken chain further opens up into the structure you see at the bottom of screen. If you were to count all the atoms, you'd see that you still have C6H12O6, so we still have glucose, but it's no longer in an alpha or beta form because it's no longer cyclical. This is called a straight chain form of glucose. All you need to note is that you now have a functional group, that means that it's an area of reactivity, around the double bond. The hydroxide group is still sitting down the bottom, but this is simply an open chain form of glucose. Note too that the functional group in green is very similar to a carboxylic acid group. Now this functional group with a double bond is reactive in a sense that it's ready to lose an electron. In other words, an oxidation reaction can occur around the double bond. Now, if the double bond is oxidizing and losing an electron, this electron can move to another species and reduce it. This means that a sugar that can do this is a reducing sugar, not because it itself is reduced, but because it can cause something else to reduce. Note again the presence of a double bond. A double bond is reactive because it's easily oxidized. Therefore, let's note a couple of things about reducing sugars. Firstly, the only sugars that can open into chain forms in solution are ones that have non-bonding carbonyl carbons. Now this means that the carbonyl carbon, which is the carbon that is bonded to both a ringed oxygen and a hydroxide group, if that carbon hasn't already reacted to form a bond with a neighboring saccharide, it can react in solution to open up the chain. This means that there is a carbonyl carbon present that hasn't been involved in bonding to form a di or polysaccharide. This means that monosaccharides like glucose, galactose, fructose, maltose, they're all reducing sugars, because if we checked out their structure, which we'll do in a moment, we'd note that they have a non-bonding carbonyl carbon. Now, this is true also of a di or polysaccharide. If a di or polysaccharide has a free non-bonding carbonyl carbon, then they are also reducing sugars, but this is quite rare. Sucrose, for an example, the disaccharide you need to know, is a non-reducing sugar as there is no free carbonyl carbon. Now let's put this to the test. There is a carbohydrate. The first thing you notice is that there's a hydroxide group bonded to a carbonyl carbon, and it's on the opposite side of the methanol group, so you have alpha glucose. Now, there is a carbonyl carbon right there. It's bonded to a ringed oxygen and a hydroxide group. What's more, it's free. In other words, it isn't involved in bonding to a neighbouring saccharide. So we give this structure a tick. It's a reducing sugar. It's got a free carbonyl carbon. Next one. You've got a carbohydrate in a ringed form. You have a hydroxide group bonded to a carbonyl carbon, but you also have the hydroxide group on the diametrically opposed carbon to the carbonyl carbon on the beta side of the ring. And therefore this is not glucose, but alpha galactose. This carbohydrate you don't need to know, but if you were given this diagram, you would need to be able to deduce whether or not it is reducing or non-reducing. So what would you do? You'd look for the carbonyl carbon. You'd ask yourself, is it involved in bonding to a neighbouring saccharide? The answer is no, therefore it's free, therefore galactose is a reducing sugar. Let's try another one. Do you recognise this pentose? Of course you do, it's the only pentose you need to know. It is fructose. But is it alpha or beta? Well, we need to find the hydroxide group. Now the hydroxide group linked to the carbonyl carbon is on the same side as the methanol group, therefore it's beta fructose. What's more, there's a free carbonyl carbon not involved in bonding to a neighbouring molecule. Therefore this is a reducing sugar. Are you getting the tricks of it? Here's another one. Now you won't know what this is by looking at it, but all I'm interested in is this reducing or non-reducing? Well, let's see if there's a free carbonyl carbon. Examining the left side of the structure, I can't see a ringed carbon that's bonded to an oxygen and a hydroxide. But on the right-hand side, I can. There's the hydroxide group, there's the carbonyl carbon, and it's not involved in bonding to a neighbouring molecule. Therefore this, which happens to be maltose, is a reducing sugar. That's an example of a disaccharide that is a reducing sugar. Here's a disaccharide that you should recognise. You need to know it, and of course at this stage you'd already know that it is indeed sucrose. And when we look at sucrose, we're looking for a free carbonyl carbon. And as you examine the structure moving from left to right, it's impossible to find a carbon that's bonded to a ringed oxygen and a hydroxide group. Because we can't find a free carbonyl carbon, we have to conclude that this is non-reducing. So sucrose is a non-reducing sugar. If you were to go backwards to see how sucrose formed via the condensation of fructose and glucose, you would see from our previous lessons that the carbonyl carbon on both structures were involved in the bonding process to form the disaccharide, and are no longer called carbonyl carbons because they have given up the hydroxide groups during the condensation process. So, if a molecule or a carbohydrate has a free carbonyl carbon, it's a reducing sugar. That's what you need to know as the Year 12 chemist. Look for the free carbonyl carbon. If there is one, it's reducing. Now we test for reducing sugars in a number of ways. There are 3 different tests that you need to be familiar with. When I say familiar with 3 tests, you would need to be able to recognise the results of 3 tests, but you only need to know the details of one test that you have actually done. Now, the test that we have done in the laboratory is the test that involves Benedict's solution. This next section of the movie will talk you through Benedict's, Fehling's, and Tollens. Here are the tests for reducing sugars. The double bond found in reducing sugars in the open chain form is the place where oxidation occurs. Now, if you have copper 2 plus ions, we can reduce them to copper 1 plus. If you had silver ions, they can be reduced to silver solid. Remember, when a species oxidizes, it is called a reductant because it is causing another species to reduce. Just think about this carefully. If a species is oxidizing, it's a reductant because it's causing something to be reduced. Alternatively, if a species is reducing, we call it an oxidant. Why? It's causing a different species to be oxidized. Therefore, reducing sugars are reductants because they themselves are oxidizing and causing another species to reduce. The first test that we need to be familiar with, at least, is called the Tollens test. Now, Tollens involves silver ions. I don't know how you're going to remember that, but the Tollens test involves silver 1 plus ions, and they're in an aqueous alkaline solution, so the pH is high, in the form of silver and ammonia bonded together. Now, if a colourless solution containing a reducing sugar is warmed with Tollens reagent, silver ions are reduced to silver solid, and they can form a silver or a black precipitate. Note that the silver is reduced by the reductant reducing sugar. In other words, the reducing sugar has oxidized around the double bond and its electrons have reduced the silver ions. Now, the reaction involved in the equation is silver ions in solution plus an electron are reduced to a silver solid. Fehling's and Benedict's tests are essentially the same thing because they both deal with a copper 2 plus ion. Now, you're used to the fact that copper 2 plus ions are a nice bright blue. If you look at the picture, the left-hand side is a solution of either Fehling's or Benedict's solution. Bright blue ions in solution. Now, if a colourless solution containing a reducing sugar, say for example a glucose solution, is warmed with Fehling's or Benedict's, the copper 2 plus ions are reduced to copper 1 plus ions. So the right-hand side test tube in the picture is an example of Fehling's or Benedict's having been reduced by a reducing sugar. The reducing sugar, we would say, has oxidized around the double bond and its electrons have reduced the copper 2 plus ions. Now, the equations are two-step in Fehling's and Benedict's. 2 copper 2 plus ions in solution are reduced by 2 electrons to form 2 copper 1 plus ions in solution. Now, the copper 1 plus ions have that copper color, but what they do is they form copper 2 oxide. This copper oxide is a precipitate that's reacted with the hydroxide groups present in an alkaline Fehling's and Benedict's solution. Copper oxide is copper colored, and therefore that's the evidence that the oxidizing reducing sugar has indeed reduced the copper 2 plus ions. So there's your tests that you need to know. Fehling's and Benedict's are copper 2 plus, Tollens deal with silver plus. Here's an example of a question involving reducing sugars from the 2008 HSC paper. It's question 33B, and you'll see that there's 6 marks allocated to this question. A first-hand investigation was performed to identify reducing and non-reducing sugars. 3 marks to describe an appropriate procedure. You've not only done the procedure in class, but you now have some slides to back up your procedure. 3 marks just to describe a procedure. 2 for 2 marks. You need to account for the results obtained in terms of the chemical differences between reducing and non-reducing sugars. You need to suggest an instrumental method for identifying different sugars, which we will cover in subsequent lessons.